Hello everyone, and thanks for listening in. My name is Fiona Backhouse, and I'm a PhD student at Western Sydney University in Australia, working on the vocalisations of Albert's lyrebirds. I'm going to talk about a study I've been doing on sequences of mimicry in the Albert's lyrebird. When animals use acoustic communication, this is often done in sequences. Sequences can include elements or syllables within songs, or at a higher order, songs within singing bouts. Sequences have been demonstrated in a number of animals, including whales and hyraxes, and particularly in birds. Sequence patterns range from singing with immediate variety, where every consecutive song is different, to singing with eventual variety, where a song type is repeated many times before switching. The predictability of the order of songs also varies between species. Some sing the same songs in the same order each time, whereas others present them more randomly. So we know that many animals use sequences with some level of structure, but for higher order sequences in particular, we still understand little about the acquisition and function of communication sequences. One animal in which acoustic sequences can be found is the Albert's lyrebird. Albert's lyrebirds are a large Ossian passerine found predominantly in rainforests in a very small area of eastern Australia, as shown in red on that map. Many of you have probably heard of the superb lyrebird, which is famous for its mimicry. The Albert's lyrebird is a sister species and also mimics the vocalisations of other species, as well as non-vocal sounds in a complex multimodal display. People listening to Albert's lyrebirds have observed them performing this mimicry in predictable sequences that may even be shared between neighbouring males. But this has never been formally quantified, so I set out to investigate these sequences of mimicry. Here is an example of some mimicry with the different species that he mimics. This study had three main questions. First, we wanted to quantitatively confirm if sequences of mimicry are in fact predictable. We then wanted to test if they're socially transmitted between individuals. Finally, we wondered what determines the order of mimicry within sequences and wanted to see if mimetic units were ordered to signal the diversity of vocalizations that an individual can sing. So I recorded the mimicry of 5 males from 5 locations, identified all the mimetic units as indicated by the different letters up here, and systematically split recordings of mimicry up into sequences based on a vocalization that every bird commonly used. I then used something called a Liebenstein similarity index to get a measure of how similar each pair of sequences is. This works by calculating the number of changes needed to make one sequence identical to another, and then standardizes this by the length of the sequences. So to illustrate, here is another sequence that is identical in its composition, so it has the same mimetic units in the same order. And here are some more sequences that are quite similar, so they share a lot of the same sequence units that are often in the same order, but they're not exactly the same. So I first compared all sequences within individuals, and I found that, on average, the similarity index between all sequences and individual male sings is about 40%. But what does this value actually mean? I wanted to compare this to the similarity you would expect by chance if the sequences weren't stereotyped. So I randomized the order of vocalizations within the sequences, and I calculated the similarities again. And I found that just based on the mimetic repertoire, you'd expect to find a similarity of about 14% if the sequences were completely random. This number is significantly smaller than what we found between the real sequences, so we can conclude that males are, in general, repeating the structure of their mimetic sequences. How does this change when you look at sequences of different males? So within males, we found the similarities of about 40%. Between males from the same location, the similarities were almost as high, close to 36%, which is still significantly higher than expected by chance. When I compared the males from different populations, the similarities were much lower, closer to 19%. However, this was still significantly higher than you would expect by chance. So this shows that sequences are largely shared within populations, though sequences might show a small individual signature. It's possible that some of the similarities might be explained by similarities in repertoire, but the permutation analysis showed that similarities in repertoire alone don't explain similarities as high as what we found. And the same model species are in fact found across the range of Albert's lyrebirds, so differences between populations will not reflect species assemblage. These results are instead strong evidence that sequences are socially transmitted between individuals. That is, lyrebirds are learning how to structure their mimicry by copying the sequences of other lyrebirds. So why are the sequences organised in a particular way, and why are there still similarities across the entire species? One of the things that Albert's lyrebirds do is sing with immediate variety, which allows them to show a high diversity of sounds within a small time frame. But why is it still so organised? I took some measurements of the units, things like frequency, bandwidth, and entropy, and I calculated the difference between consecutive vocalisations in all of these measures, and then what you might expect if they were randomly organised. What I found was that overall, the acoustic difference between consecutive units was higher than you'd expect if the sequence was randomly organised. In some visual signals, a higher contrast may mean a stronger message, such as highly contrasted colours on a poisonous animal. Here, the high contrast is probably drawing attention to the signal. We think that the mimicry of lyrebirds functions to show off a male's vocal abilities. These results suggest that Albert's lyrebirds organise their sequences to enhance acoustic contrast and therefore repertoire complexity within a short amount of time. In this study, we confirmed that sequences are an important aspect of the communication of Albert's lyrebirds. The mimicry is non-randomly organised, and sequences and mimicry are shared between local males. This social transmission of whole sequences has very rarely been shown before, and most notably in whales rather than birds. Our results also suggest two levels of learning in the mimicry. A previous study by David Putland suggested that individual mimetic units are learnt at least in part from the model species, and we suggest that the order of this mimicry is then learnt from other males. So how does this compare to song and sequence learning in non-mimetic vocalisations? We then showed that the way the mimicry is organised increases the acoustic contrast between units. Mimicry used in a sexual display may function in showing a male's vocal abilities through his ability to mimic a range of other species. This study suggested in Albert's lyrebirds it is not only the number of species of vocalisations that is important, but also the acoustic range of sounds he is able to produce. So the take-home message from this is that in Albert's lyrebirds, whole vocal sequences are socially transmitted, and the organisation of sequences is likely to be functionally important. Is this something we'll be able to find in other species? Thank you.